here now. Letters burning by my bed for you here now.
What's happening? What's happening? Oh, Benito! Well, sorry about that, everyone, but, what? What?
Hello! Hello there, and welcome back to Molecular Methodologies. Now, in the past few videos, the only organisms we've really been talking about are prokaryotes. Well, in fact, the only organism we've been talking about is E. coli. So let's liven things up a bit in this video. That's exactly what we're going to do. We're going to be talking something which isn't a prokaryote. Today, we're going to be talking about transforming eukaryotes, in particular, fungi. Like last time we talked about expressing genes in E. coli, and you might begin to wonder, why would we need to express genes in another organism when we can do it perfectly well in E. coli? Well, the answer to that question is all to do with post-translational modifications. A lot of proteins, after they've been translated, need to be modified in some way by adding certain non-protein elements to it. So I don't know, like glycolipids, for example. And that's done by an organelle called the Golgi apparatus. Now, prokaryotes don't have the Golgi apparatus, but eukaryotic cells do. So that's why we sometimes need to transform eukaryotes and not prokaryotes. And in particular in this video, we're going to be talking about yeast, Saccharomyces cerevisiae, or baker's yeast. So it's the yeast you'd use to make your lovely loaf of bread every weekend. Well, if you're a baker, that is, which some of you may be. I mean, if you are a baker, please comment below. I'm very interested in your work. Now, here's top fact of the day for you. The first eukaryote to ever be transformed was, in fact, yeast itself. And what was even more remarkable about that is that it wasn't done in a vector. So it was completely uncloned. All we got was some random piece of DNA. We chucked it at some yeast, and somehow the yeast started expressing the genes on the DNA that we chucked at it. A bloody miracle! Now, in practice that's extremely inefficient nowadays, but the reason why it worked back then was because we were using a mutant of yeast, an auxotroph. Now, an auxotroph is an organism which cannot live on or survive on a minimal medium. A minimal medium is just a basic carbon and nitrogen source. And it can't survive on that minimal medium because it can't produce a certain substance. It's got a mutation in one of its genes, which means it can't produce something which is vital for its survival. So what the scientists did, that they chucked the gene at the auxotroph that would have made it into a prototroph, an organism that could survive on a minimal medium, and then what happened, the gene that they chucked at it got integrated into the yeast chromosome. So it was now part of the genome. So therefore it could now be expressed, and we got a prototroph. Hooray! But that's the silly way of doing it. Now in this video we're going to be talking about the proper way of transforming yeast. Now, there are two types of yeast transformation vector. There's the integrative vector and the shuttle vector, which we'll come on to in a minute. Let's talk about the integrative vector. Now the integrative vector, as the name suggests, integrates itself in the yeast chromosome. And on this board, I've got to say, this is probably my best diagram of the series so far, so you know, don't thank me all at once or anything. But this is a diagram of a yeast integrative vector. So what we have is a plasmid, and let me just go through all the things that we can find on this plasmid 1 second. So we'll start up here. We have the OEE, the origin of replication for E. coli. So that's what we'd use to make hundreds and hundreds of copies of this plasmid before we insert it into our yeast cells. So E. coli is still a very good organism for doing that. We've got our BLA gene. We're very familiar with that by now. That's the gene for ampicillin resistance, the antibiotic ampicillin. So that we're pretty familiar with. That just allows us to select for transformants in our E. coli colony. Over here, of course, we've got our polylinker site where we've inserted our insert of DNA that we want to express in yeast cells. But this is the bit that's really new, the URA3 gene. Now the URA3 gene is a gene which codes for uracil. Now uracil is a base found in RNA, for example, so it's absolutely essential that the yeast cells contain or are able to produce uracil, otherwise they won't be able to grow on a minimal medium unless we supplement them with uracil. Now the strain of yeast cells which we're transforming have a mutation in the URA3 gene. Just to point out the nomenclature on this, if it has a mutation then we write the gene in lowercase, so lower URA3. What we've then got to do is make the yeast cells competent. What do we mean by that? Well, we mean that they need to be able to take up the plasmid which we're chucking at it. Now, in the olden days, we used to degrade the cell wall, the chitinous cell wall of the yeast cells, but now some clever bugger found out that if we mix the yeast cells with lithium salts, in particular lithium acetate, then that will create pores in the yeast cell membrane, which allows this plasmid to be pushed in. Once the plasmid is inside the cell and inside the nucleus, that's when the exciting stuff happens, because this is when this plasmid gets integrated onto the chromosome. Now, below here, I should have labelled this, this is the yeast chromosome. This is a section of the yeast chromosome, and here we have the mutated URA3 gene. And I've marked specifically here where the mutation has occurred. It's only a mutation of one base. So there's one base difference between the non-mutated version and the mutated version. Now this plasmid gets integrated onto this chromosome by homologous recombination. This is when two nucleotide sequences are exchanged between two very similar molecules. This occurs by this URA3 gene lining up with the mutated version on the yeast chromosome. Now after that, recombination can take place, which will mean that our integrated chromosome will now produce a functional URA3 gene which does produce uracil. Now if you think about how this works, I've tried to show it in this diagram here. So here we have the mutation here, and then this is where the plasmid combines with the chromosome here. It could be anywhere, but I've just chosen this place as an example. So then when that gets integrated, it will go all the way around there and back around here. Now at the bottom here, I've shown what the final product is, and hopefully you can make some sort of sense of this. I haven't done it quite to scale here, obviously, because there's no way I could have fitted all of this in one portrait line like that, so sorry, I do apologise. So, let's go through what we have here. Now, what we have here is the start of the original mutated uracil gene that was here. This dotted bit is this bit here. This dotted section is this bit here, which combines with the start of the mutated gene to form another non-functional uracil gene there, because there's been no correction in that mutation. We carry on, we've got our insert, that's what we're really doing this for in the first place. We've got origins of replication and our blood gene, they're not really relevant at all in this case, but here, this is where it is relevant. What we have here is the start of the UR3 gene, which was in our plasmid. This is where the mutation was in the mutated version, here. But now, we don't have a mutation, and it's stuck on to the end of the UR3 gene that was in the chromosome to start with. Now that didn't have any mutations anyway, so this part and that part combined will produce a functional uracil gene. And this is called genetic complementation. So basically, in case I didn't make that clear, this part of the gene is combining with this part of the gene, coming together, forming a functional uracil gene. And obviously if we grow these yeast cells on a minimal medium, we can select quite easily for ones that have integrated the plasmid into their chromosome, because they're the only ones that can survive. All the other ones will die because they need uracil. for their survival, and they can't produce it. Okay, so that's one type of yeast expression vector. What's the other one? It's this guy, the yeast shuttle vector. Now what's the difference? Well, instead of being integrative, so it integrates itself within a yeast chromosome, this vector is what's known as episomal. It doesn't get transported into the nucleus, it isn't incorporated in the nucleus, it just stays within the yeast cytoplasm, replicating and translating and expressing genes. Now an example of one is this one, the PS2 vector. P stands for plasmid, presumably, Y stands for yeast, E stands for expression, S stands for... You've got your usual stuff, origin of replication for E. coli, very important. You've also got your blind gene for antibiotic resistance. We're sick to death of them now. We've also got our selectable marker for use in yeast. Once again, we see our URA3 gene popping up again for uracil. But the main difference in this vector is this bit here, the 2 micron origin of replication. And the main difference between having a vector which integrates itself in the yeast chromosome and one which is episomal is that this vector actually needs an origin of replication which allows it to replicate in yeast cells. When it's integrated in the chromosome, then the origins of replication within the chromosome can do that for us. But here, this guy's all on its own, it needs to sort itself out, and that's what this 2 micron origin of replication is for. Now, it turns out that in nature, yeast cells actually have plasmids of their own. And when scientists, back in the old days, were looking down electron microscopes, looking and measuring these plasmids, they realised that they were about 2 micrometers in length. And that's what the 2 micrometers stands for. That's just a bit of background. But basically, this is a section of yeast plasmid incorporated in our bacterial plasmid, which allows the plasmid to replicate itself in the yeast cells. Now the thing about using a shuttle vector is that they aren't as stable, as you may imagine, within the yeast cells as the integrative vector. So therefore you must always be adding the selection pressure of a minimal medium without uracil. So that way only cells which have taken up the plasmid can actually produce uracil. Now the one thing we missed out is this bit here, the polylinker, where we insert our DNA. Because after all, that's what we're doing this bloody thing for in the first place. Now, here we have our insert, and either side we've got something which you may think is a bit weird, so let me just explain what they are. This, of course, is the promoter. That's where the transcriptional proteins bind and start transcription of mRNA. Now the good thing about this promoter, the p-gal promoter, is that it can be regulated. Now gal stands for galactose. That's because this promoter in nature is used for the metabolism of galactose. Now it can be made inactive when there's glucose in the growth medium, and it is induced by galactose itself. It's a bit like the lac-Z gene in a way, because remember that produces beta-galactosidase, which breaks down lactose, but that is also induced, that gene is also induced by lactose itself. So it's quite a similar thing what we're looking at here. Something that's useful because we can turn on and turn off the gene whenever we feel like. We can have a whole lot of fun with that. Last thing to talk about is the CYC1TT. Now the CYC1 isn't relevant at all. The main thing is this TT. TT, not for tiny temper, it stands for transcription terminator. Now this also reflects the differences when transforming eukaryotes compared to prokaryotes. In transcription of genes in eukaryotes, the mRNA must contain a poly-A tail. That's at the end of your transcriptional sequence, a load of A bases must be added. A load of nucleotides with A bases attached to them must be added to the end. And that increases the stability of the mRNA molecule. That's exactly what this transcription terminator provides. So when your RNA polymerase is going through the promoter, through the insert and round, it won't know when to stop, basically. So it will go through the transcription terminator and carry on going round and round. But that polyadenylation signal means that when that mRNA is being translated, it'll get chewed back to where that polyadenylation signal was. So you just get the gene that you've inserted being translated and nothing else. So that's a really good point. Eukaryotes require a poly-A tail on their RNA transcript before anything else can be done with it. So there you go, you've got your yeast, you've got your vector inside it, it's producing the gene that you want, you can extract all that protein and probably get a nice load of money out of it. Well, hey! Now in this video we've just talked about yeast, but there are other types of fungi which you can also transform, like the filamentous fungi. They're particularly famous for producing proteases which are used in washing powders. But they're a little bit less easy to work with than yeast. For a start, you cannot introduce plasmids just by introducing lithium salts. You have to break down the cell wall the old-fashioned way to create a protoplast. You usually have to use an integrative vector, not a shuttle vector like this one, because, I mean, this is pretty unstable in yeast cells, which are single-celled organisms. You have to continuously be adding that selection pressure. So in filamentous fungi, they're even going to be less stable. I'll be back soonish to talk about something even more exciting. How to transform an animal. Yes, an animal. Wow, it's going to be crazy. God, oh, I can't. I'm too excited. Look at me. Oh, God, I can't wait. I'm sorry, I'm going to have to go now. Bye. Oh, I'm so excited. Oh my God, expression of animal. Oh, I have to do a starter jump. Baka! 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 Oi, what are you still looking at? Hey, get out of it. I want my privacy here.